Okay, hi everybody. Welcome to my third hot wash. This one I wanted to take some time and talk about some transport pathways that you should probably know. So we're going to cover 8 different pathways. Our first one that we have is the carnitine transport. Then we're going to talk about the citrate transporter, the glucose alanine transporter, lipid transport, the sodium potassium pump, the aspartate malate transport, citrulline transport, and then finally the phosphorus and H plus symport protein. Okay, so starting with the carnitine transporter, this one is super important for oxidizing fatty acids. And so we're going to start off inside the cytoplasm. We're going to have an acyl-CoA group, and that is going to bind to carnitine. All right, and that's going to form our acylcarnitine. The acylcarnitine is then going to go into the matrix, where it will separate into acyl-CoA and carnitine. And then the carnitine will leave the matrix, where it can then go back into the cytoplasm to find another acyl-CoA group, and the cycle kind of repeats from there. The next one is going to be citrate transport. So this one involves shuttling acetyl-CoA out of the matrix and into the cytosol. And then that's super important to building up our malonyl-CoA for fatty acid synthesis. So inside the matrix, acetyl-CoA is going to bind to oxaloacetate. This forms our citrate molecule. The citrate then leaves the matrix and goes into the cytosol, where it will separate into acetyl-CoA and oxaloacetate. And then the oxaloacetate just goes right back into the matrix and it repeats again. Our third one is going to be the glucose alanine transporter. So this is really used for degrading muscle cells during times of really extreme starvation. It is very costly, but better than starving, and so it's really only used as a last resort. So the way it works is that the alanine is going to exit the muscle cell. It's going to go into the liver. From there, it's going to be turned into 2 pyruvate groups. And then the amino group that we had on the alanine is going to be transferred, and that's going to make glutamate. And then the pyruvate is going to go back up through gluconeogenesis, and the glutamate is going to go back to the urea cycle to dispose of the amino group. So, like I said, super expensive, right? We would really try not to break down our muscles, but if there is nothing else that we can do to get glucose, this is one way that we can do it. Next up, we have lipid transport. So, as we know, the super hydrophobic fatty acids can't simply just float through the bloodstream, so they use lipoproteins. There's kind of 4 classes. The lowest lipid content one is the high density lipoproteins. Those are responsible for kind of picking up the fatty acids and bringing them to the liver. Whereas our low density, very low density, and chylomicrons are used for delivering fatty acids. Chylomicrons are pretty uncommon. You really only see them shuttling fats from the intestines to the liver. And then from there, it's the VLDL and the LDL that's responsible for really like cellular delivery of our lipids. The sodium potassium pump is really one of the more infamous ones, right? Everybody knows it. Most people have to memorize it at least once, usually twice. One kind of neat thing about it is that it will actually scaffold with PKC, and so through like our RTK pathways, we can actually see some increased control over it. The process is pretty simple. It uses 1 ATP, it brings in 2 potassium, and 3 sodium leaves the cell. Our next one that we have is the aspartate malate shuttle. This one's pretty neat because the regular NADH isn't able to just enter the mitochondria. And so what we have to do is to form malate, which will be able to enter the mitochondria matrix. From there, the malate will actually be oxidized to form oxaloacetate. And when this happens, an alpha ketoglutarate is leaving the inner membrane, and this is exchanging for the malate. So they will switch places. The oxaloacetate is then going to go through a transamination reaction with glutamate, and this is going to form an alpha ketoglutarate and an aspartate. The aspartate then leaves the mitochondria, and there we go. You have your NADH energy delivered into the inner membrane of the mitochondria from the cytosol. This is a very complicated process because it's super important that none of this energy leaks out of the mitochondria, right? We don't want our NADH to be able to go back and forth because then we will have the risk of losing all that energy. And so it has a bunch of steps so that we can only shuttle energy in, not anything out. Okay, next up we have citrulline transport. And so what this is doing is it's working with the urea cycle. So what happens is that we're going to combine our ornithine and our carboxyl phosphate inside the mitochondrial matrix. And so because it's the only one that occurs in the matrix, we have to get our reactants in and then take our products back out. And so the way that this is happening is that the mitochondria are going to have a protein called the ornithine carrier protein. From there, the ornithine will bind to that protein and enter the mitochondria. The ORC is going to then drop off the ornithine, which will bind to a carboxyl phosphate, forming citrulline. The citrulline then binds back to the ORC and leaves the mitochondria, where it will separate from the ORC and go down into the urea cycle, which will regenerate the ornithine, and the cycle is allowed to continue. Okay, our last kind of transport one is fairly simple but very important. It is the phosphorus and hydrogen symport. And so because we need just a constant stream of both protons and phosphate groups to kind of feed the ATP synthase, what we have is we have this channel protein that will allow them to flood in, but making sure that we're not losing things so that our proton gradient's not disrupted. It's very selective to only these two molecules. And because the phosphorus is used up with the hydrogen atoms, it's going to be concentration driven so that it will actually allow the molecules to enter into the mitochondria. And that's it. If you have any questions, feel free to reach out, and thanks for watching.